time to start. Thanks for being here. My name is Brooks Miner. I'm going to tell you about work that I've been doing over the past couple of years with the freshwater microcrustacean Daphnia, which is about 1 to 3 millimeters in length, very common in freshwater food webs in temperate lakes and ponds all around the world, and very important part of lake food webs as well. So I'm going to start out showing you an environment, and the environmental variation that I'm interested in has to do with how clear the water is in terms of its transparency to ultraviolet radiation coming from the sun. So a pond in an area with a lot of vegetation around it has higher vegetation inputs, leading to higher dissolved organic matter. Dissolved organic matter absorbs UV wavelengths of light. And so ponds in this sort of a landscape are much less transparent to UV than ponds in this sort of a rocky landscape. And so here we have a cup of tea. The reason it's brown is because there is dissolved organic matter from dead vegetation, which is exactly what's going on here and what's not going on here. And this is important for organisms that live in the water because a more transparent pond has more ultraviolet radiation, and among other things, UV radiation is really important to living things because it damages DNA in a couple different ways. In previous work, we've shown that Daphnia from these more transparent ponds have higher survival under UV exposure in the lab. And what I want to talk about today has to do with digging into the mechanisms that underlie these adaptive differences in which populations from higher UV places are better at surviving ultraviolet radiation. There are 3 broad categories of ways that zooplankton respond to UV. One is to swim away from it, behavioral avoidance. This is not a key part of what's going on in the study system that I just introduced you to, partly because those ponds are really shallow, partly because some of them are so clear that even if they did swim the 1 meter depth down near the bottom, there's still plenty of UV down there. Another reason that we know that it's not the whole story is because the differences in UV tolerance that we measure in the lab occur in a situation where they don't have a behavioral refuge. Another thing that's important for lots of zooplankton is pigments that are protective against UV. So this Daphnia has melanin pigmentation in its carapace. The melanin molecule absorbs UVB radiation and presumably absorbs that radiation before it reaches the tissues on the inside of the animal. And there's been work done on that showing that organisms that have pigmentation sometimes survive better under UV experiments. Another option, if you can't swim away or shade it out, is to simply sustain damage to your DNA and then fix it. And that's one of the things I'll be talking about today. So some Daphnia exhibit this protective melanin pigmentation up in the Olympic Mountains of Western Washington, where a number of my study populations are and where these 2 photos come from. I've got how transparent the water is to UV on this, less UV to more UV axis, and the amount of melanin in the organisms. My first message for you is that up in these Olympic populations, melanin content is low. And the reason I describe these numbers as low is because the pigmented animal in the photo actually comes from the Sierra Nevada mountains, another high UV, clear water lake. And that's how much melanin a melanic Daphnia has. And this scale of variation is pretty trivial and not something that you can see when you observe them. And the trend here is actually in the wrong direction anyway. If anything, we would predict that in the clear ponds they would have more melanin. So we've got a melanin protection thing going on potentially in the Sierra Nevada populations, but not in the Olympic populations. Despite that difference, these sets of populations are closely related. We did some phylogenetic work showing that the Sierra Nevada Daphnia, which are all pigmented, are closely related to the Olympic Daphnia that are not pigmented, even though there are other populations that we included in our study that are melanin pigmented. So they're each other's closest relatives, even though they differ in this presumably important UV protective trait. So most of what I want to talk about is the repair of UV-induced DNA damage. So one of the main damage structures that's caused by UV radiation is what are called Pyrimidine Dimers. A double bond is formed between adjacent pyrimidine bases, and that causes problems down the line because the chemical structure is different, which affects the way that polymerases, among other things, interact with the DNA, so it interferes with DNA replication and RNA transcription. But lots of zooplankton, including Daphnia, have a really cool repair mechanism that we don't have, which is called photorepair or photoenzymatic repair. There's an enzyme that was actually lost in mammals called photolyase. When an organism has that enzyme and is exposed to visible and UVA wavelengths of light, the enzyme absorbs wavelengths of UVA and visible light, derives energy from absorbing those wavelengths, and uses that to catalyze a reaction that takes away the double bond. It's much less energy intensive for the organism than, for example, nucleotide excision repair, which involves cutting out the damaged nucleotides and replacing them. And so the questions that I'm going to ask and answer today involve, first of all, whether that melanin pigmentation is protective against DNA damage, because that's actually not been tested or shown before. Whether genotypes from the different kinds of UV environments, high or low, have different rates of repair of DNA damage via this photorepair mechanism. And then a question of whether they have different rates of DNA repair when light is absent and they can't do the photorepair mechanism, and they have to rely on slower and more energy-intensive processes like nucleotide excision. So to measure these pyrimidine dimers, we use an ELISA protocol with an antibody that binds to the pyrimidine dimers, and the ELISA is a colorimetric assay. We get a response back. We start the wells with DNA samples from organisms, and the darker the color we get back, the more pyrimidine dimers were present in that DNA sample. So we have genotypes that are kept as asexually reproducing clones, which is an advantage of working with Daphnia, that are kept in the lab for several generations and therefore don't have any maternal effects from their source environment. We have genotypes from a high UV pond in the Olympic Mountains, genotypes from a low UV pond in the Olympic Mountains, and then a pigmented genotype from the Sierra Nevadas. We put them all into a rig on a turntable where they're all exposed first in the dark to a high dose of damaging UVB radiation to create a lot of these dimers in their DNA. Then we turn off the UVB and we power up the visible and UVA wavelengths, allowing them to perform photorepair. And we take animals out at different amounts of time after exposure and measure how much damage there is in the DNA. Several graphs I'll show you will look like this. This is the number of hours after they got zapped and created the damage from UVB. And this is how much damage is in the DNA. I decided to just use the raw data, which is the ELISA absorbance, the absorbance of that colorimetric assay at a particular wavelength in a spectrophotometer. And the first thing I'll show you actually doesn't involve repair, it's just the initial amount of damage. So that's 0 hours of repair, so it's immediately following the damage. And what we see is that that pigmented Sierra Nevada genotype has significantly lower damage initially. And the Olympic populations have similar damage, even though some are from high UV and some are from low UV, which indicates that, our assumption that because they don't have pigmentation, they don't have differences in screening out the damage, this is consistent with that. And so now we'll look at how the repair goes. And in all lineages, they do repair the damage, but the rates of repair differ significantly, and it's a little hard to tell because they have different starting points. But we fit an exponential decay model to these data. These are the model fits for the 3 different categories. And there are significant differences in the initial amount of damage, and there are significant differences in the slope of the exponential decay curve among these 3 categories. The Sierra Nevada actually has the highest coefficient or the highest slope for repair. The Olympic population from the high UV environment has the second highest, and the Olympic population from the low UV environment has the lowest DNA repair rate, and those differences are significant. The other thing that as I said, I'm going to look at is what if we take away this photo repair mechanism? One of the things you'll see when I reveal the data is that the repair is a lot slower. So we don't actually collect the 3-hour and 6-hour time points. We just have the same initial damage, and then we have 9 hours later how much damage they have. And this is now hours in the dark. So we've disabled the photo repair mechanism simply by modifying the light environment. And now we have a linear model because we only have 2 points for each one. And again, the same initial damage data again comes out significantly different intercept. And there's a hint at perhaps different repair rate slopes. And the reason for that is that the Daphnia category from the low UV pond has essentially 0 repair in the dark. But the Daphnia from the higher UV pond and the Sierra Nevada Daphnia that are also from a high UV environment and have the pigment, they potentially show evidence of some amount of repair during this time period. I think to dig into dark repair more, I think we need to work on a longer time scale because as you can see, it's just so much slower that there's not much evidence. But this is intriguing. I hadn't looked at dark repair before, and considering that these Olympic populations are closely related to each other, and this Sierra Nevada population is more distantly related, it's a bit of a surprise that these come out looking different from each other and these have such similarity. So to sum up what I've told you about these mechanisms of adaptation to UV out there in the natural world, melanin pigmentation in the Sierra Nevada genotype is presumably protective against DNA damage. And the rates of the photorepair DNA damage are significantly different among genotypes from these 3 different kinds of UV environments, and one of which has protective pigmentation along with it. And lastly, the dark repair that occurs, although it's much slower, there's definitely an indication that we should do some more work looking at whether there are differences among genotypes and populations in that rate of repair. So with that, I would like to acknowledge collaborators, an undergrad at Cornell in particular, who I worked with a lot as a postdoc, and collaborators Jackie Lopez and Mike Frender at Notre Dame, who I've been working with for several years now. Thanks very much.